Dalo maternal class lag रही है कांत मरूंगा तो कने अगर टीवी ने
Good morning.
Hi, good morning.
Good morning, dear students. Good morning, our professor. We are glad to welcome you to our lecture. We hope that the lecture will bring you a lot of new and interesting knowledge. I think we will start. Welcome.
Okay. Can I share my screen? It's forbidden now. The host forbid the screen sharing. Excuse me? I can't share my screen. I think you need to do some setting from the host to let me share my screen. Okay, it works now. Hi guys, can you see my screen?
Yes.
Yes, sir.
Okay. My name is Wudi. It's quite easy for a Chinese name. Wu is my family name and Di is my first name, Wudi. And I'm working in Wenzhou Medical University School of Pharmaceutical Sciences and in Professor Liang's group, Chemical Biology Research Center. I was a host several years ago when your professors visit Wenzhou Medical University. So I feel quite honored today to give a report about pharmacology. And my topic is the coming age of host-defense peptides. And that is my research field. Okay, I will give you a brief introduction about host-defense peptide and some basic knowledge about amino acids and the peptides and the secondary structures of the peptides and some case study about my research. And if we have time, we can do also introduce a peptide bioinformatics. Okay. Well, first we need to know what is host-defense peptide. Host-defense peptide is also short for HDP. And traditionally, it's referred to as antimicrobial peptides, AMP. So in the following sections, I may sometimes say host-defense peptide, and sometimes I may say antimicrobial peptides. They are naturally occurring molecules that are produced as the first line of defense by all multicellular organisms. The peptides are ribosomally synthesized in our body and in nearly all multicellular organisms. And they are relatively small, less than 10,000 Dalton. And usually they are cationic and amphipathic. and of variable length, sequence, and structures. But classification of host-defensive peptide has been based on some instrument like CD, circular dichroism studies, and many of them have been supported by NMR, X-ray crystallization data, and include many like 6 major classes, like the E, which is alpha helical structures, and B and C, there are beta sheet structures with multiple disulfide bridge, and some of them with only one single disulfide bridge is looped, like B. And like F is extended, just random coil structure, and some of them are cyclic and with more complex structures. Well, most host defense peptides are broad spectrum antimicrobials. Like I just said, it's also called antimicrobial peptides. So they can kill bacteria, fungi, and viruses and parasites. And in a number of cases, their activity extended to tumor cells. They can kill tumor. And they are found in a wide variety of species ranging from bacteria through insects to human, and they can be expressed constitutively or induced in response to infections, trauma, and inflammatory mediators and cytokines. Well, like I said, is produced by all multicellular organisms like animals, insects, and plants, and fish and shellfish, and amphibians. My research area is the antimicrobial peptides from the amphibians. So let me introduce the history of the host defense peptide. Actually, the antimicrobial peptides are discovered likely in the 1980s of Bombini. You know, Bombini is from a frog, and lactoferrin from milk. And in 1981, a landmark study by Bowman reported how injection of a bacteria into a silk moth, you know, to produce the silk, to introduce a potent antimicrobial peptide. And this discovery of cyclopins from the silk moth was the first major report of alpha helical antimicrobial peptide. And another important advance was Professor Zeslov and his colleagues in 1987. They isolated and characterized the cationic antimicrobial peptide from an African frog, cloud frog. And it's called manganese. And if you do the research about this field, you must know like Paxiganum is one of the most popular antimicrobial peptide, which is in its clinical trials. And in the mid-1990s, Hoffman's group showed that the genetic ablation of antimicrobial peptide synthesis rendered the fruit fly susceptible to a massive fungal injection, and providing the first evidence of critical role of antimicrobial peptides in insect host defense. And then we discovered many peptides in mammalian skin and of course in human body, like the cathelicidin LL37. You can search it online. LL37 is quite famous as well. And subsequently, generation of mice deficient in one of the catalysides that clearly demonstrated the relevance of antimicrobial peptides to mammalian host defense. And antimicrobial peptides have thus been extensively characterized and discovered in virtually all multicellular organisms that have been studied for this activity. And present, I think now maybe there are more than 2,600 antimicrobial peptides have been deposited into the APD. You can search this database, which is called antimicrobial peptide database. It is likely that this list represents only a small fraction of gene-encoded antibiotic proteins or peptides produced in nature. And except the naturally occurring, we also to do the modifications and optimization of the peptides or de novo design of the antimicrobial peptides. So there must be more than 2,600 antimicrobial peptides in the world. Well, talking about the history of antimicrobial peptides, I have to introduce Alexander Fleming. You must know him because penicillin, right? But actually, the first reported human antimicrobial protein was even earlier than penicillin. Well, the first reported human antimicrobial protein is called lysozyme. It was identified in 1922 from a nasal mucus by Alexander Fleming. And this observation was overshadowed when in 1928, Fleming discovered penicillin, and in the 1940s, he, along with others like Channing and the other guy, and they brought the therapeutic use of penicillin to clinical, and for which he was awarded a share of the 1945 Nobel Prize for Medicine. Thus, the beginning of the golden age of antibiotics in the 1940s led to a loss of interest in the therapeutic potential of natural antimicrobial peptides, such as this lysozyme. Well, in the 1960s, the rise of multi-drug resistant microbial pathogens awakened the interest of antimicrobial peptides as host defense molecule. So here we mentioned the multi-drug resistant microbial pathogens. So we need to know what is multi-drug resistant. Like we may hear some pathogens like MRSA, it's methicillin resistant Staphylococcus aureus. That's one of the most dangerous microbial in the world. So let's see a short TED video and to see what is multi-drug resistant.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere, on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. Today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then one comes along as its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that helped them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria, like Salmonella, even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria. And although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions, such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics, such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections, can have a major impact by keeping more non-resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de-escalation may sometimes work better than an evolutionary arms race.
Well, okay, that's the emergence of multi-drug resistant microbials. So the increased frequency in the number of isolated drug-resistant pathogenic microbes in hospitals worldwide has promoted both research institutions and the pharmaceutical industries to design and develop novel drugs targeting the resistant pathogens. Well, interest has focused on host defense peptides, primarily due to their broad spectrum activity and low potential to induce resistance and a diverse range of activities. They can not only directly kill microbes, they can also modulate host immunity. And recent studies have demonstrated a wide range of activities in modulating the functions of host cells and tissues, as part of natural host defense, which has led to the depiction of these peptides as host defense peptides, HDPs, rather than antimicrobial peptides, to better define their multi-phased role in immunomodulatory mediators and, under some circumstances, antimicrobial agents. Well, like I said, except directly kill microbes, it can also have some chemotactic activity to recruit our active immunocytes to help our bodies to kill the bacteria, the invaders. And also they can modulate the TLR response, toll-like receptors response, to neutralize bacterial products to suppress inflammation. Like in the sepsis, there is an inflammation storm. Professor Liang may talked of this before. And to enhance some nucleic acid recognition to promote auto-inflammation of our body, like in TLR3 of 7, 8, and 9. I need to give a brief introduction about the antimicrobial function and the mechanism of these peptides. Let's see this bacteria. For the left section, indicated by the dashed yellow lines here, for the left section of the bacterial cell shows the mechanisms of action of antimicrobial peptides. And for the right section of the bacterial cell shows the the resistance mechanism the resistance mechanism of gram-negative bacteria. Like introduced in the video, we can see usually the microbiome could change in membrane permeability, or they can produce beta-lactamase to make the antibiotic activated. And also they can build an ion channel or some channels to exflux the antibiotics. So all the mutations need to happen in the proteins. But we see the membrane of microbiome is only bilayer, right? The phospholipid bilayer. And the bilayer could not be changed too much. So that's also the advantage of antimicrobial peptides to kill the bacteria, because they usually works on the phospholipid bilayer and then kill the bacteria. So the bacterial cell could not change too much on the bilayer to resist antimicrobial peptide. So let's see how the peptides work on our microbes. Well, the ability of the antimicrobial peptides to kill bacteria usually depends on upon their ability to interact with bacterial membrane of cell walls. Yes, there is another cell wall beyond the membrane, the cell membrane. So generally, the peptides exhibit a net positive charge. We say the cationic antimicrobial peptide, so it's a net positive charge and a high ratio of hydrophobic amino acid to allow them to selectively bind to the negatively charged cell membrane. And the polyanionic, which means the negatively charged microbial cell surface, usually contains acidic polymers such as lipopolysaccharide, LPS, in gram-negative bacteria, and lipoprotein teichoic acid in gram-positive bacteria. They are all negatively charged. So there is a electricity attractions between the peptides and the cell membrane. And the transit of the outer membrane of this peptides could also onto the surface of the bacterial cell membrane, and the self-promoted uptake was happened. And why the antimicrobial peptides could have some selectivity? That's because the specific species, due to the difference in the membrane composition of different microbes, and also from the microbes and human cells. Like human cells is also always neutral, the electricity, and the bacterial cells is always negatively charged. So the antimicrobial peptides could kill the bacteria, but without harming our human cells. And that's the selectivity of these peptides. And after interact with the cell membrane, subsequently, these peptides will insert in a manner such that they initially straddle in the surface of the hydrophilic head groups and the fatty acyl chains of membrane phospholipid. After insertion into the membrane, antimicrobial peptides act by either disrupting the physical integrity of the bilayer via membrane thinning or transition and disruption of the barrier function. Or sometimes some antimicrobial peptides could translocate across the membrane and act on some internal targets in the cytoplasma. Several complex and conventional models describe these subsequent events, including the reorientation of peptide molecules. They insert into the membrane perpendicularly and to form either barrel-stave or toroidal channels, like here, the barrel-stave models or toroidal models. Then breakdown of the membrane integrity as a result of the swamping of the interface or detergent-like dissolution of patches of membrane, and the formation of the membrane charged by a coupled of peptide lipid aggregates within the bilayer. Each of these successfully predicts the ability of cationic antimicrobial peptides to break down the cytoplasmic membrane, but only the toroidal channel and aggregate model explain the action of certain peptides on cytoplasmic targets. Indeed, the action of many peptides cannot be explained just by disruption of membrane permeability barriers. Other highly charged peptides may also interfere with the activity of the cell wall, lytic enzymes on the outside of the bacterial cell. And that's the antimicrobial function mechanism of the peptides, directly killing the bacteria. And we just said it can also modulate host immunity to modulate the immune response and regulating inflammation. Like the peptides can act as chemoattractants that could initiate the recruitment of innate immune cells to the infection site and initiate cytokines induction by viral cells in some chronic autoimmune diseases. The cytokine-induced catalysidins, like IL-37, could express and overexpress of human defenses, which is also a HDP, confers protection against skin infections, whereas the anti-inflammation nature of antimicrobial peptides can also suppress both chronic and acute inflammatory conditions by influencing the toll-like receptors, TLRs. The TLRs could recognize the microbial products and nucleic acid released upon the tissue damage. So one of my research now is the LPS neutralizing peptides, which could neutralize LPS in our human body to cure the sepsis after infection. Well, actually, there is already evidence about the immune modulations by using the next generation sequencing. Here is the complex response of mouse macrophage to human beta defensin, one of the host defense peptide in human body, to human beta defensin 3. For the left panel is a network interaction diagram that shows the proteins belonging to the interleukin signaling pathways and the lipid and the lipoprotein metabolism pathway. And these are a subset of genes that changed the expression after treatment with human beta defensin 3, as determined using GO2R126. Although these pathways accomplish diverse functions within the cell, they are interconnected and share several common proteins. And for the right panel is the summary of the expression patterns of cytokine signaling genes in macrophages activated by endotoxin, RPS alone, or RPS together with HBD3. Although cytokine signaling is thought to be dampened by the presence of the HBD3, many genes belonging to this pathway are upregulated. And the network diagram and the expression data were generated by using network analyst 8. Here are the immunomodulation of host defense peptide, HBD3. And actually for our human body, diversity of HDP activities within the body and relationship with the disease states. Host defense peptides are produced by various different cell types throughout the body. Here are HDPs that are produced by the epithelial cells of skin and lung and gut, and some inflammatory cells in circulatory system. On the skin, certain HDPs such as the catalysidin, which I talked about, the IL-37, and human beta defensin, HBDs, and human neutrophil peptides, HNPs. They are constitutively expressed by keratinocytes and mast cells, whereas the expression of other can be strongly induced in response to injury or infection, which attract immune cells to the area surrounding the damaged tissue. Well, in the lung, HDP is produced by airway epithelial cells help to protect from invading microorganisms. Well, in the gut, plant cells produce large amount of human alpha defensin 5, HD5, and HD6 at the base of intestinal crypts, helping to prevent infection from pathogenic bacteria and maintain homeostasis of the commensal microorganism community. Well, in the circulatory system, many immune cells express HDPs in response to infection and inflammation. These HDPs can have multiple effects throughout the body, depending on the location where they are produced and the types of cells that are present. A dysregulation of HDP expression at any of these sites can contribute to various disease states. So now we have introduced the host defense peptide. So what exactly has affected the different functions? Here are the peptides we just mentioned, like HMP, alpha defensin, beta defensin, HBD3, and IL-37, the human cathelicidin. So we can see that the structure, the different structure may affect the different functions. So let's go to some basic knowledge about amino acids and peptides. First, let's recall the what's the name? A central dogma of molecular biology. Still remember? Well, inside the nuclear is DNA, and DNA is actually quite long. For human, the DNA, if you can measure the length, the DNA strand may be 2 meters length. And each strand of the DNA holds the code of create specific proteins or peptides. And because the DNA could not leave the nuclear, so they need to transcript to the mRNA, the messenger RNA, and the mRNA could translocate to the outside of the nuclear. And mRNA takes this information, the genetic information outside of the nuclear, and bring it to the ribosome. The ribosome moves along the mRNA, reading the code, and this phase is called translation. First with transcription, and then the translation. And another type of RNA is called tRNA, transfer RNA, collects the specific amino acids that are needed to make the protein. And the tRNA brings the amino acid to the ribosome, and the ribosome will build a chain of amino acids. A chain of amino acids, that's the polypeptide chain. And based on the code in the mRNA, it's called elongation. And then the ribosome continue to move down the mRNA strands until all the appropriate amino acids are added and the protein is complete. That's how the peptide chain or protein was made in the cells. And if we denature the normal protein or peptide, we can get the denatured peptide chain. That's the basic sequence. The primary structure of a peptide chain is the sequence of this peptide. And we can look deeper, and we can see the basic blocks are the amino acid. So do you still remember the basic structure of amino acid? Why is it called amino acid? Because it's an acid, right? We call the organic chemistry knowledge. It's an acid. Here is an acid group. acid group. So it's generally called acid, but this acid is with the amine group. So it's called amino acid. And it is actually called alpha amino acid because the amine group is connected with the alpha carbon. It's called acid. So the carbon near the acid group is called alpha carbon. And the amine group is connected to the alpha carbon. So it's called alpha amino acid. And there are another two groups, which is hydrogen, and the other one is side chain. And the side chain can differ from each other. So we got different amino acid. Okay, like in the central dogma of molecular biology, we already know the peptide chain elongation. Well, the hiding reaction actually is a condensation reaction. The chain's two amino acid molecules can be covalently joined by a peptide bond to yield a dipeptide with the elimination of water, and this is called condensation. Here is the acid group, and here is the amine group. And after the condensation reaction, we got a peptide bond, and that is a dipeptide. And the reverse reaction is also called hydrolysis. We need a molecule of water to hydrolyze this dipeptide into two amino acids. So we can see the amino acids is very important. It's the basic blocks of peptides. We can use amino acids as building blocks. Different organisms can make extremely diverse products. Even in one human body, we can make enzymes, hormones, antibodies, and transporters, and hair, muscle fibers, and even the lens protein of the eyes. And for some birds, the feathers, and for the spider webs, they are all proteins or peptides with distinct biological activities. So there are infinite possibility of amino acids are commonly found in proteins. Asparagine was the first amino acid discovered, and the last to be discovered, the 20th, was threonine in 1938. Well, for the 20 different amino acids in peptides synthesized on ribosomes, we can classify them with different methods. Like we can classify them into essential and non-essential and conditionally essential amino acids. Essential amino acids must be consumed in the diet, and non-essential is, of course, can be synthesized in the body. And conditionally essential is cannot be synthesized due to the illness or lack of necessary precursors. For example, the premature infants, they always lack of sufficient enzymes needed to create histidine. So histidine for premature infants, they are essential, actually. And also we can classify them by the property of the amino acids. Like we can see here, amino acids with electrically charged side chains, like arginine, histidine, and lysine. They are positively charged. For the research of antimicrobial peptides, these R, H, and K, arginine, histidine, and lysine, they are quite important, especially arginine and lysine. They are very popular in host defines peptide. And usually when we do the modifications, because we need the peptides are cationic, positively charged. So sometimes we will do the alteration of one amino acid into the other, and we use the positively charged amino acid R and K. And some negatively charged, like aspartic acid and glutamic acid, D and E. These are charged amino acids, and also uncharged, but it's polar, like serine, threonine, and asparagine and glutamine. They are polar but uncharged. And also we have some hydrophobic side chains. So they are unpolar and uncharged. They're hydrophobic, like the tryptophan, tyrosine, and phenylalanine. Still, we have some special cases like glycine and proline and cysteine we may introduce later. So you may need to know the different classification of the amino acids. Well, common structural features I already introduced before is they are alpha amino acids, and except proline, because proline later we can see there is no free amine chains actually, and alpha carbon is bonded to 4 different groups, except glycine, because glycine the side chain is also hydrogen, so there are 3 different groups, right? A carboxyl group, an amine group, and an R group side chain, and a hydrogen atom. And the alpha carbon atom is a chiral center. So there is an enantiomer of this this amino acid actually. For the human body, in the proteins or peptides, there are always L stereoisomers. So there's L stereoisomers. There must be R stereoisomers, exclusively L stereoisomers in our human body, and the D amino acid residues. L amino acid to make the L stereoisomers, and D amino acid residues could also make the D stereoisomers. You know, some peptide chains are unstable against the enzymes. So sometimes we use D amino acids to replace the L amino acids to make sure the peptides could be resistant to the enzymes. And we already introduced the R group for the classification of amino acids. You see the proline. Proline is one of the special amino acid because it's a cyclic structure. Here is proline. Pure proline. There is a 5-atom proline. And the polar uncharged R groups, positively charged and negatively charged. Here, you can recall this later. And cysteine, we talked about cysteine. Cysteine in our 20 antimicrobial peptides is quite important because 2 cysteine could form the disulfide bridge, disulfide bond. And disulfide bond is quite important in the secondary structure, like we talked about the beta sheet. Beta sheet structure is formed by the disulfide bond. And actually in human body, there are uncommon amino acids. Here are 6 of these uncommon amino acids. It can also form the peptides or proteins in the human body, but they are uncommon amino acids. For now, there are over 300 has been discovered. It could increase or decrease protein's activity. Some of the amino acids are the free amino acids. Some of the uncommon amino acids are synthesized to the peptide chain or protein, and then do the post-translational modification, and they form the uncommon amino acids. And the peptides could also classified into oligopeptides or polypeptides. Oligopeptides consist of only a few amino acids, always less than 10, whereas polypeptide chains consist of a longer chain of amino acids. Like this amino chain, how many amino acids does it have? 1, 2, 3, 4. 4 peptide bonds, so it's formed by 3 amino acids. Formed by 5 amino acids, so it's called pentapeptide. If 2 amino acids to form a peptide is called dipeptide, and tripeptide, and tetrapeptide, and pentapeptide, and hexapeptide. These are all oligopeptide. And usually the molecular weight of below 10,000 is called polypeptide, and above 10,000 is usually called protein. Well, I just introduced the central dogma, but maybe not clear. So here there's another small video to show you how the peptide chain was produced in our human body.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, this is the biosynthesis of peptide chain in our human bodies. But if we want to develop the host-defense peptide into drugs, we can not only just extract the peptides from human bodies or from the animals, because there are too small amount of the peptides. So how can we get enough peptide? Well, for larger proteins, we can, of course, we can use protein engineering, tissue engineering to fertilize something like that. But for our short peptide, actually, we can use chemical synthesis. But it's a little bit different from the normal organic chemistry. The normal organic chemistry always is liquid phase synthesis. But for peptide chains, we always use SPPS, solid phase peptide synthesis. Well, generally, you need to know the protecting group. We always use pharmacochemistry. We use pharmacochemistry to protect the amine group because a free amine group is quite active and we need to protect it first. And then we can use a base like piperidine to remove the red ones, which is pharmac, the protecting group. And then we got a free amine group. And then we can start the synthesis. Like the yellow color here is actually resin. Small particles, material like sand is a resin. And on the resin there is a amine group. Always, but sometimes other groups. But we take amine group as an example. After the deprotection of the first step, we got a free amine group on the resin, and then we do the coupling. Here's the amine group. Here is the acid carbonyl group. So we can do the condensation reaction, right? To remove molecule of water, and we got the peptide bond, NHCO, the peptide bond. And now we have the first amino acid here. And the amine group is still with another protecting group. And then we do the deprotection step again. And we got another free amine group. And we do the coupling of the second the second amino acid. And then we got another peptide bond. And now it is a dipeptide. And we deprotect the protecting group, and we got another free amine group. And we do this round and round to get further coupling and deprotection circles to get a longer peptide chain. Now I think we can do the solid phase peptide synthesis less than 100 amino acid. I think that this is possible for nowadays. And the last step is to cleavage step to cut down the peptide chain from the resin. And the peptide chain could dissolve in DMF or DCM, and the resin can be filtered. And you see here, the cleaved peptide chain. Here is the N terminal, and here is a amide group, and we can make it to acidic group as well. So this is C terminal, and this is N terminal. So we can see the difference of chemical synthesis of peptide chain and biosynthesis of peptide chain. Well, for the chemical peptide synthesis most commonly starts at the C terminal, the carboxyl end of the peptide, the C terminal, and then proceed towards the N terminal, the amino terminal. And for the biosynthesis, is occurs in the opposite direction. And you can see from the reaction, and Y is from the C terminal to N-terminal. And for the carboxyl group, actually this condensation reaction is not quite easy to be happened. So always we use a catalyst like HOBT or HOAT to form an active ester in the intermediate state and to catalyze this coupling reaction. If you have interest about SPBS, you can do some further work. Well, this introduction about the peptides, and we can also show you some knowledge about the secondary structures. Well, we talk about the primary structures that's made by the amino acid residues. The sequence is actually the primary structures. And then it could be assembled and to form secondary structures. Like what we introduce most is alpha helix. And the secondary structure could form tertiary structures and quaternary structures to form larger proteins. For our host-defensive peptide, we usually talk about the secondary structure. It's the stable arrangement of amino acid residues giving rise to recurring structure patterns. Well, because of time, I may eliminate some pages. Well, here is the structure of a host-defense peptide. The known three-dimensional structures of proteins and peptides, actually it can achieved in the PDB, Protein Data Bank, PDB. You can search this website. And this image shown here was made by using the data from the entry with PDB ID 2L24. It is a HDP, host-defense peptide. And some important features of protein or peptide structures. The function of a protein or peptide depends on its structure, right? And the most important force stabilizing the specific structure maintained by a given protein are non-covalent. And what is conformation? You need to know some definitions. The spatial arrangement of atoms in a protein or peptide is called its conformation. And proteins in any of their functional folded conformations are called native protein or native peptides. And here is also definition of motifs. That's major for the tertiary structures. Well, a protein's conformation is stabilized largely by weak interactions. So what is weak interactions? We already said the disulfide bond. The disulfide bond is actually a covalent bond, so it is a strong interaction. But the weak interaction, there are hydrogen bonds and hydrophobic effect and ionic interactions. For the disulfide bond is, you can see the Gibbs free energy is quite large, so the covalent bond is a strong bond, like the disulfide bond. For the hydrogen bond is quite useful for the secondary structure of a peptide. It's partially electrostatic attraction between a hydrogen atom, which is bound to a more electronegative atom or group, such as nitrogen or oxygen. In the peptide chain is also always nitrogen and oxygen. They are the hydrogen bond donor, and the other hydrogen is a hydrogen bond acceptor. So they can form a hydrogen bond. There is this weak force. So for proteins and nucleic acids containing NH or OH group, they always form hydrogen bond. And for alpha helical conformation, the secondary structure, is mainly because of the hydrogen bond. You see the hydrogen bond here to help the peptide chain to form alpha helical conformation. And sometimes we also have some electrostatic interactions. So that's it's called salt bridge or ionic bonding. Here is what we do here. You can see the interactions between our peptide and the protein. There are many ion bridge to stabilize the function. And the van der Waals force and the hydrophobic interactions. We see the antimicrobial peptides are always hydrophobic, so the hydrophobic interactions to maintain its secondary structure is also very important for HDPs. Well, the predominant secondary structures are alpha helix and beta sheet, and also beta 10, and some undefined, also called random coil. Here is the alpha helix. It looks like this alpha helix. The right-handed alpha helix is a common form, right-handed, but sometimes we have a left-handed secondary alpha helix. And actually we can do some modifications to make the peptide to be a left-handed alpha helix to resistant to the enzyme hydrolysis. Left-handed and right-handed. And this amino acid sequence could actually affect the stability of the alpha helix. Like we talked about the proline and glycine, they are quite special, right? Because proline, the nitrogen atom is a part of a 5 atom rigid ring, and the rotation about the nitrogen and alpha carbon bond is not possible. So it's usually to introduce a destabilizing kink in alpha helix. So it's always the kink or the start of alpha helical conformation of a peptide chain. And for glycine, it's different as well, because glycine has more conformational flexibility than the other amino acid, because the side chain is only a hydrogen, quite small and quite flexible. So the proline and glycine, they are always the start of alpha helical conformation. So when we do the modification, we usually change the proline or glycine into other amino acids. And some constraints that affect the stability of alpha helix, the intrinsic propensity of amino acid itself, and the interaction between the R groups, and the bulkiness of adjacent R groups, and the occurrence of proline and glycine residues. So that's the constraints that affect the ability of alpha helix. Why we talk so much about alpha helix, that is because most of the host-defense peptides are alpha helical peptides. Well, but some still some other peptides are beta sheet conformation. Well, you can see the beta sheet conformation by yourself. Is you need to know what you need to know is there is anti-parallel beta sheet and parallel beta sheet, a little bit different. That's the introduction about amino acid and the peptide and the secondary structures. So let's see a case with what is my research achievement. Well, like I said, I do the research about the antimicrobial peptide from frog skin, from the amphibians. So here is a novel antimicrobial peptide biopeptide from the amphibian skin secretion. We discovered this few years later, and we published this paper last year. It's called dermoseptin, because it's found on the skin secretion, so it's named to dermoseptin. And it is also some research model for this kind of field. So we can give you a brief introduction to introduce this field. The frog we use is Philomedusa salvageis from South Africa in the Amazon jungle. And this frog, you can search it on YouTube, is quite interesting. Well, frog skin, we get the frog skin secretion from it without harming the frog. And then we do the molecular cloning by using the frog skin secretion. Because in the secretion, there are the peptide and there are the mRNAs. So we can extract the mRNAs and do the molecular reverse transcription and do the molecular cloning. Then we get a full sequence of different peptide. And then we found the peptide, which is a novel. Nobody found it before. So we name it as dermoseptin PS2. And we also need a HPLC, the chromatography, to further to give some further evidence that it is really occurred in the frog skin secretion. So we do the HPLC, and we found it's actually a complex chromatogram in 240 minutes was provided by the crude skin secretion analysis performed by the reverse-phased HPLC. And following the MSMS, the mass spectrometer, we can identify the peptide is actually exist in the skin secretion. And then we do the dichroism, circular dichroism CD to identify the secondary structure, and we found it is actually the alpha helix structure. And then we do some molecular modeling to get our 3D model of this peptide, and it is the alpha helical structure. And then we do some activity screening because we want to see the antimicrobial activity. So what we do is to do the MIC and BC, which is minimal inhibitory concentration and minimal bactericidal concentration assays for the peptide. And 5 key species of microbes, including gram-positive bacteria, both sensitive and resistant, Staphylococcus aureus, we talked about before, and MRSA is methicillin-resistant Staphylococcus aureus, and also gram-negative bacteria, E. coli and Pseudomonas aeruginosa, which is always from biofilm, is also resistant to many kind of antibiotics. And also we pick the Candida albicans, a yeast, to see the antimicrobial peptide activity against the fungi. And we found minimal concentration, inhibitory concentration, and the bactericidal concentration is quite low. So the activity is good. So later we do the minimal biofilm inhibition concentration and the biofilm eradication concentration and the initial attachment assay to see if our peptide could inhibit or eradicate the biofilm that formed by this kind of bacteria, and we found the anti-biofilm activity is also quite good. So next we will check the mechanism under the good activity. So we do the peptide localization experiment by observing the FITC labeled peptides using confocal microscopy. And all the bacterial cells could be stained with green color by the cyto9, it's a dye, while the bacteria with disrupted cell membrane showed the red color by staining with the dye PI. And comparing to control, the permeability of the cell membrane of MRSA was significantly affected by the treatment of our peptide at the MIC concentration, because many bacterial cells captured in the red color. Okay, and besides, the bacterial cells displayed permeabilized membrane at MBC concentration, since most of the bacteria were stained with red color, MIC concentration and MBC concentration. And that's the evidence of our peptide works on the cell membrane and disrupt the cell membrane. And furthermore, we use scanning electron microscopy to investigate the effect of peptides on cell membrane. You see, the control was observed with a smooth and intact surface and completely separated. That's the control. That's Staphylococcus aureus, and here is the gram-negative E. coli. And you see that's the control, quite beautiful bacteria. It's smooth and intact surface. Well, the normal cells were evidently lysed, followed by fusing, clumping, and disintegration by treating with our peptide for 1 hour. And abundant cells clustered was evidently observed in the environment. And also for the, it's not E. coli, it's Pseudomonas aeruginosa. It's incubated with our peptide displayed aggregation, shrinking, and collapse. Both the planktonic bacterial cells and the adherent cells embedded in the biofilm had been damaged in the presence of our peptide. And later, it was found that our peptide molecules was spread on the surface of gram-negative bacterial membrane, adopting alpha helical conformation by our molecular docking result. And during our investigation, we found another interesting thing. Well, the rigid symmetric multimer docking showed 2 peptide molecules could form an approximately anti-parallel form, and the hydrophobic phase is exposed to the solution rather than being buried inside in the dimer interface. It's quite different. So we hypothesize that the hydrophobic peptide may have some self-assembly tendency. So we do the investigation. We use DRS, dynamic light scattering assays, and we found after the incubation of 3 hours and 24 hours and 72 hours, the particle size is quite large compared with the fresh peptide solution. And we also use microscopy to see the particles that the self-assembled peptide incubated for 72 hours. So later we may see if there are any connections between the self-assembly activity and the antimicrobial activity. We also do some in vivo activity test, like the efficacy of this peptide on infected skin wound healing was evaluated by a marine skin MRSA infection model. The treatment of this infection model was assessed by the validation of the proliferation and stable bacterial viable count. And this peptide could potently inhibit the MRSA growth in the infected epidermis. And the variable count result of peptide treatment group display significantly decrease compared to the control group, which is only the salt water. On day 1 and after the 3rd day treatment, the variable count remained at a low level, and the wound surface start to scab with shrinking compared to the control group that failed to control the growth of MRSA. The peptide treated infected wound was efficient to decrease the bacterial viable count and showed distinct wound healing activity. Well, that's one of the peptide we are doing research for now. Here is the whole procedure of our research. We do the molecular cloning, and by using multi-TOF MS or SQMS to get new antimicrobial peptides from the skin secretion of Philomedusa salvagea, the frog. And the peptides are identified, and we got its secondary structures by circular dichroism and molecular simulation. And then we found the cationic peptide exhibited significant broad spectrum antimicrobial activities, and also against the biofilms. And actually, we do the safety experiment to see if it's toxic to mammalian red blood cells, and we found it is quite safe to use. And then we do the in vivo evaluation of the peptide, and we found it has potent efficiency of the treatment of MRSA infected murine skin model. And furthermore, what beyond our expectation is that the MSPS2 could display a self-assemble tendency. So we can use this tendency to develop more antimicrobial peptide materials, actually. And we also see what behind the antimicrobial peptide activity. So we do the mechanism study. And we hope the defensive peptide could be a good drug candidate in the future. Okay, that's the case of our antimicrobial peptide investigation. Well, the last part is peptide bioinformatics. Because of time, I can show you the website, and if you have interest, you can start it by yourself. It's quite easy, actually. You can search it like the NCBI, you must know the website from the NIH. And on the right side, you can see BLAST, B-L-A-S-T, which is basic local alignment search tool. And on the right panel, there is a protein BLAST. You can search the peptide sequence and see the information. Input the sequence and see the information, description, graphic summary, alignment, and taxonomy. Here is the alignment of this peptide. And here you can see the related information, structure, 3D structure. You can display it on the website directly. Here is the 2L24 we mentioned before. 2L24. And if you click, you can see more information about the antimicrobial peptide, even the reference and author's title journal. And you can directly click here and search the paper in PubMed. But if you want more structural information, you can go to the PDB, Protein Data Bank, to search the PDB ID or the sequence or the name of your peptide, and you can see more information. Like the 1224, you can see the information here. And then if you want to do some local research about the structure, you can download the FASTA sequence or PDB format. PDB format, you can open it by PyMol or some other softwares. The PyMol is free for the educational institutions, so you can download the PyMol. And open the PDB format, then you can see the information about the structure and do some modifications and to make beautiful figures. Like this. This is produced by PyMol. And also, if you do it online, you can do some change, like shown in cartoon or spacefill or line. There are also other very useful website you can check, like in the NPS, you can do some secondary structure prediction, GOR4, you can search it to see if your protein is alpha helical or beta sheet protein. And if it is alpha helical, you can do some deeper analysis by using this HeliQuest. And also, this website is quite important. And you can check the molecular weight or the number of amino acid or theoretical PI and the composition of your peptide. And later you can use your PDB file to do the rampage to see the Ramachandran plot analysis. And this is the plot is used to validate your protein or peptide structure is useful or not. Okay, guys, that's the end of my report. We show you the coming age of host-defense peptide, and we already know the importance of our host-defensive peptide and the basic knowledge about amino acid and the peptide and the proteins, and also the secondary structures and my own research project. So hope you enjoy my report and hope you can enjoy your summer school, and hope to see you maybe later. Okay, that's all.